Welcome to the deep dive, where we take these complex topics and really break them down into essential, digestible knowledge.
That's the goal.
Today, we're tackling one of the most fundamental processes in all of biology, how living cells actually power themselves.
Yeah, it's central to everything.
We've got this great chapter you sent over from Campbell Biology in Focus, all about cellular respiration and fermentation. Our mission today: cut through the jargon, get to the core insights.
It's a huge topic, absolutely, but it's also incredibly elegant when you look closely.
How so?
Well, think about it. Every single thing a cell does, building stuff, moving around, even just sort of existing, needs energy.
Right.
And that energy for almost all life on Earth, it traces back to the sun, right?
Phew.
Flows through the ecosystem.
And then the cells grab it.
Exactly. They harness it. So we're going to trace that whole journey right down to the molecule so you can really get why these processes are just so vital.
Yeah, like how does a puffin use the energy from a fish it just ate? Or how do we use the energy from lunch to, we'll do this right now?
Precisely.
This deep dive is all about explaining how cells harvest that chemical energy and turn it into a form they can actually use, mostly ATP.
The energy currency.
And we want to make it feel tangible, without needing diagrams. So let's unpack this. Where do we start?
Well, first things first, cells need energy because they're constantly working.
Makes sense.
And the main way they get it is through what we call catabolic pathways, basically breaking down complex organic molecules like sugars or fats.
Okay, breaking things down.
And the source highlights two main strategies: fermentation,
Mhm.
which happens when there's no oxygen,
Anaerobic.
right? And aerobic respiration, which needs oxygen. We'll focus mostly on aerobic respiration because it's way more efficient. Think of it like a really controlled, slow burn inside the cell.
A controlled burn, I like that, not just an explosion.
Exactly. And the key mechanism here, what makes it controlled, is electron transfers. We call them redox reactions.
Redox. Okay, oxidation and reduction.
You got it. When something loses electrons, it's oxidized. When something gains them, it's reduced. Remember, oil rig, oxidation is loss. Reduction is gain.
Ah, yes, I remember that from chem.
It's the same idea. So think about food molecules like glucose. They're packed with hydrogen atoms, and those hydrogens carry electrons with high potential energy, like water behind a dam.
Hilltop electrons, the source called them.
Good analogy, yeah. When these electrons fall towards something really electron-hungry, like oxygen, energy gets released.
Then not all at once, like setting gasoline on fire.
Precisely. That's the crucial part. If it happened all at once, the cell would just, well, fry.
So how does it manage that release? Slowly.
In steps, a whole series of steps, and that's where electron carriers come in. The main one to know is NAD+.
NAD+.
Right. Think of it like an empty taxi. It picks up two electrons and a proton from the food molecule, becoming NADH.
So NADH is the taxi with the passengers, the
Yeah.
energy.
Exactly. It's a fully charged energy shuttle, holding on to that energy, ready to drop it off later in the process, nice and controlled.
Okay, that makes sense. A controlled release using these shuttles.
Yep. And this whole process, aerobic respiration, is laid out in three main stages, or acts, as you said.
Right, the three acts, what are they again?
First is glycolysis. Happens out in the main cell fluid, the cytosol. It literally means sugar splitting.
Splitting glucose. Okay.
Second, assuming oxygen is around, we move into the mitochondrion, the powerhouse.
Ah, the famous one.
That's where you have pyruvate oxidation and then the citric acid cycle, or Krebs cycle. This stage finishes breaking down the fuel, releasing carbon dioxide.
Okay, furnace mode.
Kinda, yeah. And the third act, the big finale, is oxidative phosphorylation. This also happens in the mitochondrion. It involves the electron transport chain and something called chemiosmosis.
And that's where most of the ATP is made?
That's the big payoff, yes.
Yeah.
Almost all of it, actually. It's important to distinguish, though, a tiny bit of ATP is made directly in glycolysis and the citric acid cycle.
How?
It's called substrate-level phosphorylation. An enzyme just directly transfers a phosphate group to ADP. Simple transfer.
Okay, like a direct deposit. Small amount.
Very small. But nearly 90% comes from oxidative phosphorylation, which is powered by those NADH electron shuttles we talked about. It's a much more complex and high-yield process.
Got it. Substrate level is simple and direct. Oxidative is complex and the main event.
You've nailed it.
All right, let's dive into Act 1, glycolysis. Sugar splitting in the cytosol.
Right. Takes one molecule of glucose, which has 6 carbons.
C6H12O6. Still remember that one.
Huh. Good. And it splits it into two molecules of a three-carbon compound called pyruvate.
Okay, 6 carbons in, 2 times 3 carbons out.
Exactly. Now, what's really fascinating is how ancient this process is. It doesn't need oxygen, and it happens in the cytosol outside of any complex organelle,
Meaning?
meaning it likely evolved really early in life's history, probably before Earth's atmosphere had much oxygen, and before cells evolved complex structures like mitochondria. It's almost universal.
Wow. Okay, so how does it work? Is it just one step?
Oh no, it's a sequence of about 10 steps, but we can think of it in two main phases. First, the cell actually has to invest some energy.
Invest. It spends ATP.
It does. It spends two ATP molecules to destabilize the glucose and get it ready.
Okay, got to spend money to make money, huh?
Sort of, because the second phase is the energy payoff. In this phase, the cell makes 4 ATP molecules and also loads up 2 of those NADH electron shuttles.
So spend 2 ATP, make 4 ATP.
Net gain of 2 ATP.
And you get 2 NADH shuttles loaded up too.
Exactly, per glucose molecule. It's not a huge energy yield, but it's quick, it doesn't need oxygen, and it produces that pyruvate, which can then go on to yield much more energy if oxygen is present.
Okay, so glycolysis nets 2 ATP and 2 NADH and gives us 2 pyruvate molecules. What happens to pyruvate now?
Well, that depends. If oxygen is available, those pyruvate molecules get actively transported into the mitochondria.
Into the powerhouse.
Right. And just inside, before the main cycle starts, each pyruvate undergoes a little conversion step. It's oxidized.
Meaning it loses electrons.
Yes, and one carbon atom is snipped off and released as carbon dioxide CO2.
The stuff we breathe out.
That's part of it, yes. The remaining 2 carbon fragment then attaches to a helper molecule called coenzyme A, forming something called acetyl-CoA. And in this conversion, another NADH shuttle gets loaded up.
Okay, so pyruvate becomes acetyl-CoA, we lose a CO2 and gain an NADH, and that happens twice, right? Because we had 2 pyruvates from 1 glucose.
Exactly right. So from that initial glucose, we've now generated a total of 4 NADH, 2 from glycolysis, 2 from pyruvate oxidation, and netted 2 ATP. And we have 2 molecules of acetyl-CoA ready for the next stage.
Which is Act 2.
Act 2 proper. The citric acid cycle, also known as the Krebs cycle. This is where the rest of the original glucose molecule gets fully oxidized.
The metabolic furnace, you called it.
Yeah, it fits. The acetyl-CoA enters the cycle by joining with a 4-carbon molecule already present, forming a 6-carbon molecule, citrate, hence the name, citric acid cycle. Then through a series of 8 enzyme-catalyzed steps, this citrate molecule is progressively broken down. Carbons are stripped off and released as CO2.
More CO2 we breathe out.
Correct. And crucially, electrons are harvested, loading up more energy shuttles.
How many shuttles per cycle turn?
For each acetyl-CoA that goes in, the cycle generates 3 NADH molecules, 1 molecule of another carrier called FADH2.
FADH2, another shuttle.
Yep, slightly different, carries electrons at a slightly lower energy level, but still important. And the cycle also produces the equivalent of 1 ATP molecule directly through substrate-level phosphorylation again.
Okay, so per acetyl-CoA, 3 NADH, 1 FADH2, 1 ATP, and releases 2 CO2.
Perfect. And remember, we started with 2 acetyl-CoA from our 1 glucose.
Right, so double all that for the original glucose molecule. By the end of the citric acid cycle, we've gotten, 6 NADH, 2 FADH2, and 2 ATP from the cycle itself.
Plus the NADH and ATP from glycolysis and pyruvate oxidation.
So where are we now in total energy capture?
We've netted only about 4 ATP directly so far. 2 from glycolysis, 2 from the cycle. But we've loaded up a lot of electron carriers. A total of 10 NADH and 2 FADH2 per glucose.
Wow. Okay, so most of the energy isn't ATP, and it's stored in those NADH and FADH2 molecules.
Exactly. They're holding the vast majority of the energy extracted from the glucose. They are the high-energy messengers carrying their precious cargo to the final stage.
Act 3. Oxidative phosphorylation, the grand finale.
This is where the real magic happens, the big ATP payoff. It has 2 connected parts, the electron transport chain and chemiosmosis.
Okay, let's break that down. The electron transport chain first.
Imagine a series of protein complexes, like little machines, embedded in the inner membrane of the mitochondrion. It's like a molecular bucket brigade.
Okay.
NADH and FADH2 arrive and drop off their high-energy electrons at the beginning of this chain.
Handing off the hot potatoes?
Kind of. As the electrons are passed from one protein complex to the next, they move to slightly lower energy levels. Think of it like them tumbling down an energy staircase.
And energy is released at each step.
Small, manageable amounts of energy are released at each transfer. Now, what happens at the very bottom of the staircase? Who catches the electrons finally?
Oxygen. You mentioned it earlier.
Bingo. Oxygen is the final electron acceptor. It's highly electronegative, meaning it strongly pulls those electrons towards it. It combines with the electrons and some hydrogen ions, protons, to form water, H2O.
So that's why we need to breathe oxygen to pull electrons down this chain.
That is fundamentally why. Without oxygen to accept those electrons at the end, the whole chain backs up, like a traffic jam. NADH and FADH2 can't unload their electrons, and ATP production grinds to a halt.
Okay, so the chain was electrons downhill towards oxygen, releasing energy. But how does that energy make ATP? You said it wasn't direct?
Right. This is the clever part. The energy released as electrons move down the chain isn't used to make ATP directly. Instead, the protein complexes use that energy to act as proton pumps.
Pumping protons. Hydrogen ions.
Exactly. They pump H+ ions from the inner compartment of the mitochondrion, the matrix, across the inner membrane into the narrow space between the inner and outer membranes, the inner membrane space.
So they're building up a concentration of protons in that space.
Precisely. They create a steep electrochemical gradient, lots of positively charged protons crammed into a small space. This gradient represents stored potential energy. It's called the proton motive force.
Like water building up behind a dam, you said.
That's the perfect analogy. You've got this built-up pressure, this force, wanting to flow back down its gradient, back into the matrix.
And that's what drives ATP synthesis.
Yes. Through the second part, chemiosmosis. Embedded in that same inner membrane is an amazing enzyme complex called ATP synthase.
The molecular motor.
It really is. Our source describes it like a rotary motor or a water wheel. It has a channel that allows the H+ ions to flow back down their gradient back into the matrix.
Following the pressure.
And as the protons flow through ATP synthase, they literally cause part of the enzyme to spin, like water turning a turbine. This spinning motion provides the energy for ATP synthase to grab ADP and inorganic phosphate, pi, and stick them together, synthesizing ATP. Lots of it.
That's incredible. So the electron transport chain creates the proton gradient, and the flow of protons back through ATP synthase drives ATP production.
That's oxidative phosphorylation in a nutshell. The chain oxidizes the fuel via NADH, FADH2, and the phosphorylation of ADP is powered by chemiosmosis.
So how much ATP do we get from this whole process, start to finish? One glucose molecule.
The theoretical maximum is often cited around 30 or 32 ATP molecules per glucose. It's not an exact number because there's some minor variables like how efficiently the NADH from glycolysis gets its electrons into the mitochondrion, and sometimes the proton gradients used for other tasks too.
Still, 30-ish ATP compared to the net 2 from glycolysis alone. That's a huge difference.
Massive difference. Cellular respiration using oxygen is incredibly efficient at extracting energy. The book says about 34% of the energy in a glucose molecule is converted to ATP.
34%. How does that compare to, say, a car engine?
Oh, much better. Car engines are maybe 20–25% efficient at converting gasoline energy into movement. The rest is lost as heat.
So cells are pretty good engineers. What happens to the other 66% of the glucose energy?
It's released as heat, which isn't necessarily waste, especially for organisms like us. That heat helps maintain our body temperature.
Right, keeps us warm. Okay, that covers aerobic respiration. But what if there's no oxygen? You mentioned fermentation.
Right. If oxygen isn't available to act as that final electron acceptor, the electron transport chain backs up. NADH can't unload its electrons.
Traffic jam.
Exactly. And if NADH can't be recycled back to NAD plus data, then even glycolysis will stop, because glycolysis needs NAD plus to accept electrons in its payoff phase.
So the cell needs a way to free up that NAD plus if oxygen isn't around.
Precisely. And that's the main point of fermentation. It's an anaerobic process, no oxygen needed, that regenerates NAD plus from NADH by transferring the electrons to an organic molecule, usually pyruvate or a derivative of it.
So fermentation itself doesn't produce ATP.
No, the only ATP you get under anaerobic conditions via fermentation comes from glycolysis itself, that net 2 ATP. Fermentation is really just an accessory process to keep glycolysis running by regenerating NAD plus trap.
Okay. What kinds of fermentation are there?
The book highlights two common types. First is alcohol fermentation. Here, pyruvate is converted into ethanol, the alcohol in beer and wine, in 2 steps. The first step actually releases CO2.
Is that the CO2 that makes bread rise?
That's exactly it. Yeast does alcohol fermentation. We use it for brewing, where the ethanol is the prize, and for baking, where the CO2 bubbles are the key.
Okay. And the second type?
Lactic acid fermentation. In this pathway, NADH transfers its electrons directly to pyruvate, reducing it to lactate or lactic acid. There's no CO2 released here.
And where does this happen?
Certain fungi and bacteria do this. It's used to make cheese and yogurt. And importantly, our own muscle cells can switch to lactic acid fermentation during really strenuous exercise.
When you're sprinting and can't get enough oxygen to your muscles?
Exactly. Your muscles need ATP faster than your blood can deliver oxygen. So they rely on glycolysis and use lactic acid fermentation to regenerate the NAD plus needed to keep glycolysis going for that quick burst of energy.
Is lactate what causes muscle soreness the next day?
That used to be the thinking, but current evidence suggests lactate itself isn't the main cause of delayed onset muscle soreness. It gets cleared out pretty quickly. The soreness is more likely due to microscopic muscle damage and inflammation.
Interesting. Okay, so fermentation gives you 2 ATP via glycolysis, while aerobic respiration gives you up to 30 or 32. Big difference in energy yield.
Huge difference. Cellular respiration, by completely oxidizing glucose using that electron transport chain and oxygen, extracts far, far more energy.
Now, we focused entirely on glucose,
Yeah,
but we eat other things. Proteins, fats. Do they fit into this picture?
absolutely. The pathways we've discussed, especially glycolysis and the citric acid cycle, are like major metabolic interchanges or crossroads. They don't just handle glucose.
So other foods can feed in.
Yes. Other carbohydrates, like starch or sucrose, are easily broken down into glucose, or intermediates of glycolysis. Proteins are broken down into amino acids. Their amino groups are removed.
Deamination.
Right. And the remaining carbon skeletons can enter glycolysis or the citric acid cycle at various points, depending on the amino acid.
And fats. People always say fats are high energy.
They are indeed. Fats are broken down into glycerol and fatty acids. Glycerol can be converted to an intermediate in glycolysis. The fatty acids are broken down through a process called beta oxidation.
Beta oxidation, what does that do?
It chops the long fatty acid chains into 2 carbon units, which enter the citric acid cycle as acetyl-CoA.
Ah, straight into the furnace.
Pretty much.
Yeah.
And because fatty acids have such long carbon chains with lots of CH bonds, they hold a ton of high-energy electrons.
So that's why they yield so much energy.
That's exactly why. A gram of fat yields more than twice as much ATP as a gram of carbohydrate. They're incredibly energy-dense fuel.
Makes sense. And it's not just about breaking things down for energy, right? These pathways provide building blocks too.
That's a crucial point. Metabolism isn't just one way. These pathways are hubs. Intermediates from glycolysis and the citric acid cycle can be siphoned off and used as precursors to build other molecules. The cell needs amino acids, fatty acids, components for nucleotides. It's called anabolism.
So catabolism breaks down, anabolism builds up, and these pathways are central to both.
Beautifully put. It's a highly regulated, incredibly versatile system that allows cells to manage energy flow and build complexity.
Wow. It really is an incredible journey, from a sandwich down to electrons flowing through molecular machines to make ATP.
It's fundamental to life. Seeing how cells meticulously extract that energy step by step, avoiding that chaotic explosion, it's really elegant.
We've covered those electron carriers like NADH, the amazing proton motive force, like that dam.
Yeah.
That spinning ATP synthase motor. It really connects the microscopic world inside our cells to the grand flow of energy through the whole planet.
It absolutely does. From sunlight captured by plants to the chemistry powering your thoughts right now, it's all connected through these pathways.
So next time you take a breath or even just move a finger, maybe give a little thought to that incredible dance happening inside billions of your cells.
The constant conversion of fuel into life.
Well, I think that wraps up our deep dive for today. Thank you so much for walking us through that.
My pleasure. It was great exploring cellular respiration and fermentation with you. Hopefully this breakdown makes these essential processes a bit clearer, maybe even more engaging.
I certainly found it helpful. From all of us here at the Deep Dive and the Last Minute Lecture team, thank you for listening.
Keep digging deeper.